Welcome back to the class, children. In today's class, we are going to discuss about gene interaction. Right? Interaction, that is acting among themselves. Right? The genes interact among themselves, thereby affecting a phenotype. We know each and every phenotype is controlled by genes. Right? So here, so far we have discussed about the Mendelian concepts. Right? According to Mendel, a particular phenotype is controlled by a particular gene. Right? So what Mendel said is each and every character is controlled by one gene. But here what we are going to study is the alleles of a single gene or the alleles of different gene interact among themselves and they control a phenotype. This is entirely different from the concept of Mendel. Mendel, what he informed us, the alleles of single gene is responsible for producing the dominant or recessive characters. But here, the alleles of a single gene interact among themselves, or the alleles of different genes interact among themselves to produce a particular phenotype. Here, what is spoken about is the genes are interacting. We can say like this, it is the functional association between the genes. The genes are associated or genes work together, they function together to bring about a particular phenotype. That is what we call it as a gene interaction, right? And it may be the alleles of the same gene or it may be the alleles of the different gene, right? So they interact among themselves or with the alleles of the different gene and bring about the phenotype. So the interaction of alleles of gene or genes to control a phenotype is said to be the gene interaction. So basically two types of gene interactions are there. First one is said to be the allelic gene interaction or intragenic gene interaction, and the second one is said to be the non-allelic gene interaction or intergenic gene interaction. Let us discuss one by one. Allelic gene interaction, which is also called as intragenic gene interaction. As the name indicates, intra means within the same, right? Here, the alleles of a single gene, right? We have earlier discussed each gene has a minimum of two different alleles, two different versions, right? But there are many cases the alleles are more in number, right? So there may be 2 or 3 or even 4, right? So there are different forms of alleles of a single gene, right? So different alleles of a single gene interact among themselves, right? And produces the phenotype or affects the phenotype, right? So the interaction between the alleles of a single gene, right? So that is what very important, right? That is why it is called as intragenic. Intragenic means within the same gene, that is alleles of the same gene, right? Interaction between the alleles of the same gene is said to be the intragenic gene interaction, which is also called as allelic gene interaction, right? Let us discuss about this through an example, right? So look at this image. So this is a homologous chromosome and this is a pure type. Both are homozygous dominant, and these alleles are responsible for producing the color of the plant, and the color of the plant is red. Since red is the dominant flower, it is denoted by the letter capital R. So this is a one plant variety producing the red color flower, which is the dominant, right? At the same time, this is another plant of the same species producing white colored flower, which is recessive, right? Recessive to the dominant red color. So hence it is a recessive. It is denoted by the letter lowercase r. This is normal. When these two were crossed, when these two were mixed, right? According to Mendel, either red has to come or white has to come, right? So since red is the dominant, red has to come in the F1 generation, right? Right? But if we cross these two things, right? What is the product what we got in the F1 generation is pink, which is a combination of both the dominant and recessive character, right? So here look at this chromosome. We have the genotype has capital R and small r, right? Both the alleles are present, right? But if it follows the Mendelian rule, because of the presence of this capital R, the flower must be a red color one. But it does not follow the Mendel's concept, right? Here, a new phenotype is obtained. How this new phenotype is obtained? That is because of the interaction between these two alleles, right? Because the alleles of these two alleles of the same gene interact together, right? So they are producing certain chemicals and they interact in such a way, thereby producing a new phenotype, right? And if you notice this image, right, the image clearly depicts that it does not follows the Mendelian law of inheritance, right? Mendel states when a pure breeding homozygous dominant is crossed with a pure breeding homozygous recessive, right? So when these were crossed, a dominant form is crossed with a recessive form, only the dominant form will appear in the F1 generation. But if you notice here, it is not the dominant form, it is an intermediate form which is produced, right? Here the F1 hybrid has both the genes, right, the dominant form as well as the recessive form. Both the alleles are there, but the dominant allele is not expressed, right? Recessive allele is also not expressed. Instead of these, a new phenotype is obtained, and this phenotype is because of the interaction between the alleles of the same gene, right? So this is what we call it as intragenic gene interaction, that is interaction between the alleles of a same gene, right? So these two alleles are involved in the production of the color of the flower, and they are present on the same locus, right? So the gene is present in the same locus, and the alleles of the same gene interact among themselves, thereby producing a phenotype, right? So this is what we call it as intragenic gene interaction. The next type of gene interaction is intergenic gene interaction, right? So this is another type. So this is about the interaction of alleles between genes which are located away, right, or between the different genes, right? Intergenic gene interaction, right? So let us see this through an example, right? So this is a homologous chromosome, right? So these were the two alleles of a gene, right? These alleles were responsible, right? These alleles, capital W, is responsible for producing a white colored fruit, right? So the dominant allele, capital W and capital W. So these were the two alleles which are responsible for producing the white colored fruit. The recessive form of this allele, the small w and small w, is responsible for producing the green colored fruit, right? So first there are two forms of alleles. The dominant allele produces the white colored fruit, whereas the recessive allele is responsible for producing the green colored fruit. But if you notice this one, the second image, there is a recessive condition, right? The small w's were there, but the fruit is of different color, yellow color. The green color is not expressed, right? But what we have said, the dominant form W is responsible for white color and the recessive form W is responsible for green color. But even though the recessive condition is here, the color is not green. It is yellow color. How this fruit has become yellow color? And this is because of another dominant gene, what is called as G, which is located at some other loci on the same chromosome, right? So the color producing gene is located at two different loci. So this is one gene which is responsible for producing the color, and there is another gene which is also responsible for producing the color, but it is located at some other position, right? So two genes, right, two different genes has a direct impact in producing the color, right? And that is why even though the recessive condition W is present, small w is present, the color is not green. Why? Because there is another dominant gene what is called as capital G, and this capital G is responsible for producing this yellow fruit. Whereas if you notice this once again, in this condition we have the dominant gene for W and also the dominant gene W for white color and also the dominant gene capital G for yellow color. Both the dominant genes are present, but only the white is expressed. What about the another dominant gene capital G, that is the yellow colored gene? So here this gene or these alleles of this gene suppresses the effect of this dominant gene, right? So that is how they are interacting, right? So the alleles of this gene W, so W suppresses or it inhibits the activity of the capital G, thereby preventing the capital G to produce its yellow colored fruit. So these two dominant genes were interacting, right? So white suppress the effect of G and the product is white. Here if you notice this is already recessive, small w, green, but this is dominant. Here this dominant G suppresses the effect of this green color producing allele. So this yellow color suppresses the effect of green color and the product is yellow. Whereas if you notice this chromosome, both the alleles were in recessive condition. Under this condition only the green color gets exhibited. So here the alleles which are located at different loci or the genes which are located at different loci of the chromosome interact in such a way affecting the phenotypes, producing different types of phenotypes. That is what we call it as intergenic. The interaction between the alleles, the interaction between the alleles of different genes. Though this is one gene located in one locus of a chromosome and this is another gene located at another locus of the chromosome, right? Two genes, both are involved in the production of the color, but they are different genes located at different loci, right? So the alleles of these genes interact together and impact the phenotype. That is what we call it as intergenic gene interaction, the interaction between the alleles of different genes located at different loci of the chromosome, of the same chromosome. Different loci of the same chromosome is said to be the intergenic gene interaction or non-allelic gene interaction, right? If you take both the type of gene interaction, whether it is a intergenic gene interaction or intragenic gene interaction, in both the gene interactions we cannot expect the Mendelian law of inheritance or Mendelian rules. Most of the Mendelian's ratios we can't expect, right? That is why these type of gene interactions comes under the non-Mendelian inheritance pattern, right? So all these gene interactions does not follow the Mendel's result, that is 3:1 ratio or 9:3:3:1 ratio. All these ratios cannot be expected because all these ratios are the Mendelian's ratio, because according to the Mendel, one gene is controlled by, one character is controlled by one gene, but here the alleles interact among themselves in most of the cases, in most of the organisms and in most of the characters. Most characters are produced because of the interaction of different alleles of same gene or different gene, right? So all these gene interactions comes under the concept of non-Mendelian pattern of inheritance, right? They do not follow most of the Mendel's ratio, right? So these were the two different types of gene interaction, intragenic and intergenic gene interaction. So one such gene interaction what we are going to discuss is the incomplete dominance, which is a type of intragenic gene interaction, right? Incomplete dominance, right? We have already discussed while studying about the monohybrid cross, when two pure breeding varieties were crossed, only one form will be expressed in the F1 generation, and the character which is expressed in the F1 generation is said to be the dominant, and which is not expressed is said to be the recessive, right? This is according to Mendel. That is why the F2 ratio comes as 3:1. But here, this is a deviation of the Mendelian's dominant law, and here it is given the name incomplete dominance, right? And this was first studied by Carl Correns in the year 1905 in Mirabilis jalapa, right? 4 o'clock plant, Mirabilis jalapa. This is a very common plant in India, and in this plant he performed this experiment, the same monohybrid experiment what Mendel has performed, but the results were entirely different from the Mendel's ratio, right? Now let us see what had happened in this incomplete dominance, right? Here the Mirabilis jalapa produces two different flowers, right? So one plant produces flowers which are red in color, another plant produces flowers which are white in color. Two different plants producing two different colored flowers. When these two plants were crossed, according to the Mendel, either the red has to come or the white has to come. Any one form has to be obtained. This is according to Mendel. But here in Mirabilis jalapa, what Correns obtained is a new variety or a new phenotype, the pink color, which is an intermediate between the parental forms, right? So here that one parent has pure red color flowers, another parent has pure white color flowers, whereas the F1 hybrid is a intermediate color between the two parental varieties, that is pink color. That is why he assigned the phenotype as capital R1, capital R1, here also capital R2, capital R2. Usually we used to denote the recessive forms indicating the letter, the lower case. But here neither is recessive. This is also dominant and this is also, we cannot say which is dominant, right? The red is also not the dominant and the white is also not the dominant, right? So both are interacting, right? Both the alleles of the two different plants are interacting, thereby producing the new phenotype, right? So this is denoted as capital R1 R1, and this is denoted as capital R2 and R2, and the genotype of the F1 hybrid is R1 R2, right? So here a new phenotype which is an intermediate between the two parental varieties. Then as expected, he performed the self cross, right, between the F1 hybrids, right? So the F1 hybrids were self crossed, but what he had obtained in the F2 generation is he obtained both the parental forms as well as the intermediate forms, right? But in the ratio 1:2:1. Here look at the ratio, both the phenotypic as well as the genotypic ratios are same. See, one thing in incomplete dominance what we have to understand is, if the genes were interacting and producing the new phenotype, when this phenotype is selfed, right, because of the interaction of parental varieties, the new phenotype is obtained. But when this new phenotype is selfed, it produces the intermediate phenotype as well as the parental phenotype. What is understood from this is, only the characters are blending, only the phenotypes are blending, mixing, not the genes or the alleles, right? That is why we can able to get the pure breeding red as well as the pure breeding white in the F2 generation, right? So only the phenotypes are blending, not the alleles, right? That is why it is said to be the blending of characters, not the blending of genes or alleles, right? So here, because neither white nor red is dominant, it is said to be a incomplete dominance, right? So incomplete dominance, we get an intermediate form of both the parents. An incomplete dominance shows the ratio 1 is to 2 is to 1. That is both phenotypic ratio. If you look at this image, the red color flowers are produced in one fold, whereas the pink color flowers are produced in two folds, and the white colored flowers are produced in the one fold, right? So the ratio, the phenotypic ratio is 1 is to 2 is to 1. At the same time, if you look at the genotypic ratio, the same 1 is to 2 is to 1. So both phenotypic and genotypic ratio of the incomplete dominance is same, 1 is to 2 is to 1. This is a deviation of Mendelian ratio. In Mendelian ratio, the phenotypic ratio is 3 is to 1 and the genotypic ratio is 1 is to 2 is to 1, right? Whereas here we get the phenotypic ratio as 1 is to 2 is to 1. This is entirely different from the Mendelian ratio, right? That is why we call these gene interactions as the non-Mendelian inheritance pattern, right? So this is about the incomplete dominance. So in incomplete dominance, when two pure breeding varieties were crossed, and the product will be an intermediate form of the two pure breeding varieties, right? So and the ratio will be 1 is to 2 is to 1. So hope children, today we have discussed about what is gene interaction, the different types of gene interaction, and one of the gene interaction, incomplete dominance. Hope this session will be a useful one. Let us meet in the next episode. Thank you.